So where are we? We are attempting to talk about, or we started talking about translational modification. Basically what that means is translation, the final product of translation is a polypeptide. But that polypeptide, in order to be called a protein, needs to be properly folded and functional. Right? So the folding is important, and we finished talking about the folding. So now let's talk about how this protein can be functional. So just merely being properly folded doesn't mean that this protein does anything. Okay? So that's, and then improperly folded protein, as well as unwanted proteins. For instance, a protein that was made a month ago is worn out, and the cell has to recycle the protein. So these proteins are targeted for ubiquitination, and then will be targeted for the proteasome. A question came up yesterday where somebody asked about, what about mutated proteins? Why aren't mutated proteins digested? Mutated, just by a protein being mutated because of a genetic mutation doesn't mean that this protein is misfolded, right? Those are two completely different things. Misfolding means that the problem is in the folding. Okay, does that make sense? So don't lump misfolding with a mutated protein. Okay. Even for proteins that are formed, for instance, there are polypeptides that are pulled into the ER, and some of them are soluble proteins, but now they're misfolded or they're unwanted. Then they have to be taken out. These proteins in the ER lumen have to be taken out into the cytosol and then marked by ubiquitination and be delivered to the proteasome. Why is this misfolded protein, why does it need to be transported out into the cytosol? Because that's where the proteasome is. Okay. Just want to see if you're paying attention. Okay. So that's folding. Very important. The next type of modification is called covalent modification, and we talked about this, and we'll talk a lot more about this. And this is covalent modification is a means to control or regulate protein function. Okay? So a protein might be made, and a protein can be modified, okay? And the modification can either activate or inactivate a protein. Okay? And a protein can be activated and inactivated multiple times back and forth through covalent modification. So there are a lot of different types of covalent modification. We'll talk about a few. We already talked about acetylation, okay? But we'll talk about a few more. But in the ER, so proteins, there are certain proteins that have to undergo what is called glycosylation. Glycosylation only occurs in either the rough ER or the Golgi apparatus. Those are the only two places. So even if a protein were a soluble protein and not a membrane-bound protein, the polypeptide still needs to be pulled into the ER, because this is where glycosylation occurs, right? This is one of the function of membrane-bound organelles is to categorize or to box in functions, right? Specific functions. So if there's a specific function somewhere, then molecules have to be delivered there. So the specific function is glycosylation, and it only occurs in the rough ER or the Golgi. Therefore, that's why polypeptides have to be delivered to the ER. Okay? Glycosylation is the addition of carbohydrate residues. And I say carbohydrate residues is because carbohydrates can come into different length and different complexity, and you can have the addition of carbohydrates onto the protein, but then you can have addition of carbohydrates onto the existing carbohydrates on the protein as well, right? Not just to the protein itself, but onto the existing carbohydrates that's already in the protein. So we refer to all of that as glycosylation. Not only that, but you might have existing carbohydrates and then certain pieces get broken off. So you have the addition and removal of carbohydrates. So we include all of that under glycosylation, okay? And that is a major type of covalent modification. One thing to remember, in the cytosol, okay, not cytoplasm, but cytosol, there are no glycosylate. The protein that has undergone glycosylated are called glycoproteins. There are no glycoprotein in the cytosol. Glycoproteins only exist if it were soluble. It has to be secreted into the extracellular matrix, or if it were membrane bound, it has to point out toward the surface of the cell. But there are no glycoproteins in the cytosol. Okay, very important. We'll go through this again later on. Phosphorylation is another type of covalent modification. Is the addition of a phosphate group. A phosphate group that comes from mainly ATP, right? So adenosine triphosphate. So there are 3 phosphate groups. Okay, so that's what a phosphate group looks like. And so phosphorylation is the addition of a phosphate group. And phosphorylation can occur in many different places in the cell. Isomerization is the formation of a disulfide bond or linkage. And remember, within a tertiary structure, definitely within a quaternary structure, that disulfide bonds have to be formed. Okay? And then there are other types of covalent modification, acetylation, like we said. Ubiquitination is a type of modification, right? So think about a misfolded or unwanted protein that is ubiquitinated. That means that there's covalent modification on it, and then that tells the cell what to do with that protein. Okay. So when we talk about we did talk about extracellular matrix. When we say that there's sugar gram substance, but also there are proteins that are in the extracellular matrix. Many of these proteins are glycosylated. Okay, this is just a pretty picture to show the point. No need to memorize that. Okay, so that's covalent modification. Simple enough. Polymer assembly. So there are certain proteins that they are called proteins. So they're not in the they don't form a quaternary structure. They're just a tertiary structure. There are certain proteins that, when they're made, they really don't have any function. Right? The individual subunit doesn't have a function. However, when these proteins bind to one another and form a polymer, then the polymer itself has a function. For instance, your hair, keratin, right? Your hair is made up of subunits called keratin. Keratin itself doesn't really do much, but when keratin is added together, now you have hair. Or keratin is the subunit for intermediate filaments, which is a type of cytoskeleton, right? So keratin doesn't do anything inside the cell, but when it forms an intermediate filaments has a lot of functions. Okay, so the polymer assembly or polymerization, the process of assembling polymer, right? So polymerization is a way to regulate, is a translational modification. So you're not adding anything to the protein, but if these proteins interact with one another, that's modification, then the whole polymer has a function. Individually, each protein doesn't have a function. Okay. Which is, again, we're not talking about quaternary structure, so this is not the case where, for instance, hemoglobin is not an example of polymerization. Right? Hemoglobin just needs 4 subunits that have to form a quaternary structure. We don't call that a polymer, right? We just call that a quaternary structure. That's why there are 2 different names. Okay, so polymerization is a type of translational modification. Yes, sir.
Oh, so when you make the protein subunits, the way they connect together do they have to have like the right, say like the bonds and stuff?
Yes, they have to
So-
have binding sites,
Okay.
affinity, right. So, and we'll talk about this more. So we keep saying about specificity and binding and binding sites. What makes one protein have affinity for another? What are these? I need the structural detail. What is important for my right hand to be able to grab a hold of my left fist?
What's the word?
Size, shape,
Right.
and charge.
Okay.
Size, shape, and charge, right? Binding has to do with size, shape, and charge.
Okay.
If you don't have the right size, but you have the right shape and charge, no binding. If you have the right shape and size, but not the right charge, no binding, right? So all three have to be there.
Okay.
Now, size, shape, and charge, depending on how well how big the size is relative to the other thing, how good a shape is in matching with the other shape, and how many charges actually coordinate has to do with weak or tight binding. Okay, so you don't have to have perfect size, shape, and charge, but it has to be close enough. So whenever I say affinity, high affinity versus low affinity, specificity, it has to do with size, shape, and charge. That's all. And that's fundamental chemistry, right? You learned that in 4th grade somewhere, I think. Right? That is very fundamental. No retake. So this is another example of the collagen, the same. Here's collagen drawn this way in a ribbon, and then collagen drawn this way. Okay? So again, just pretty picture to show the point. The 4th type of modification is called proteolytic modification. So let's say, and this actually has to do with exon as well. Okay, so here's the explanation of what proteolytic modification first, and then let's talk about exons, and let's talk about alternative RNA splicing. So you can't get away from that, okay? So a polypeptide is made, and it's actually folded, okay? But it's inactive. It's inactive. So in order to activate this protein, to make it active, then translational modification or processing has to occur. And in this case, the modification is the removal of this middle piece, right, this gray piece right here. So then there's an enzyme called a protease, not a proteasome. A proteasome is a whole complex. A protease is, the example is an elastase, is a protease, and chymotryptase. The protease will cleave here and cleave here, eject that, and so now you have this. So that's proteolytic modification. You cleave off, you remove a piece of the protein, right? And now this is active and this is not. This example of insulin. A better example is actually of during the coagulation process where blood coagulates, a lot of these clotting factors, right? They are secreted into the extracellular matrix in a what we call a pro form, right? So they are in the active form. They're in the bigger but inactive form. They're in an inactive form but a bigger form. By allowing this protein to be active, you have to undergo proteolytic modification to remove a part of it, to make it smaller. Okay? So bigger isn't always better. So in order to activate this protein, it has to become smaller. And how it becomes smaller is by, again, proteolytic modification. Okay? And if you, let's say, if you lack the enzyme that carry out, if you lack the protease that carry out proteolytic modification of clotting factors, guess what? You're not going to have clots. And so it's possible that somebody who has a disease or a disorder where you don't clot very well, you call bleeders, right? So bleeders, for the most part, some of them would have a deficiency in these proteases, for instance. Okay? So that's proteolytic modification. function. All right, so this is size, shape, and charge. I previewed you on this. So why modify a polypeptide? To get it in the right size, shape, and charge. That's what it is, right? You have a polypeptide that folds, but it doesn't have the right size, shape, and charge to do whatever it is. So now it has to have the right size, shape, and charge, which we refer to as structure, right? And if you have the right size, shape, and charge, then it has the right function. So structure, function. Okay. So that's so we'll look at some examples of how modification like binding, for instance, polymerization, like covalent modification, like proteolytic modification. How do those activities lead to variation in size, shape, and charge so that the protein can have the proper function? Does that, you understand where I'm going here? Okay. So, and I can't say size, shape, and charge enough. I'll say it another 10,000 times, 10,001 times, okay? So, structure, function. Unfolded protein, you don't have a binding site. Folded protein, you have that pocket of binding site, right? So that could be a pocket of, might call that a binding site. For instance, transcription factors might have a DNA binding site, okay? Is that different from a catalytic site? Yes and no. What is a catalytic site? A catalytic site is a site where the enzyme must bind to the substrate to do something to the substrate. So it's still about binding. Right? It's still about binding. Does that make sense? Okay? So binding, right? So you control enzymatic activity, you control protein activity by controlling the structure, which then correlates with function. That's the point. That's the point of this slide. Okay? So an example. You have a membrane protein, a transmembrane protein. That means that there's a domain on either side of the membrane, right? And then it crosses through. And this membrane protein is a sodium-potassium pump or transport protein. But it transport protein, but it doesn't have a lumen, right? It's not empty in the middle. It's actually solid. So what happened is that this right here, right, has to grab the ion, the sodium from this side, stuff it in here, and then open up to release it. Okay, because it's not empty in the middle. Does that make sense? So then, number one, two questions. How does this sodium potassium pump open up in either side, right? Number one, and number two is what actually determines whether the pump is in this conformation or that conformation. And by the way, conformation is the same thing as structure. Okay? So it all has to do with binding here. So normally, this transport protein would have multiple binding sites, and it has three that has the right size, shape, and charge to bind sodium. Right? When one sodium molecule binds, not much happens. When the next sodium molecule binds, not much happens. When the third sodium molecule binds, that causes conformational change, right? Conformation of what? Of that protein, whereby it is recognized by the enzyme that catalyzes phosphorylation, or which is called a kinase. A kinase is an enzyme that catalyzes phosphorylation. So in this conformation, a kinase doesn't recognize this pump. In the conformation where three sodium ions bind, right? Then the pump has a different conformation where kinase now can phosphorylate the pump. Phosphorylation, what does that cause? That causes further conformational change, leading to that. So when a pump that is unphosphorylated is like this. a pump that is phosphorylated is like this, right? So how do you think that the pump needs to be this way again? You have to remove that pump, that phosphate group. That's the idea. So what is the function of covalent modification? Conformational change. It's all about conformation. It's all about structure. And it's all about structure leading to function. And that's why the first set of exam, we talked about conservation of function, conservation of structure because of that. Because structure and function always go hand in hand. So now when in this conformation, the conformation of the binding site for sodium ion changed, thereby ejecting the sodium ions. At the same time, the conformation for the binding of potassium ion also changed now, is able to bind to potassium ions. So that's what happens.
Okay.
When the potassium ions bind, that causes conformational change of the entire pump, that causes dephosphorylation or the removal of the phosphate group, which is carried out by an enzyme called a phosphatase. So kinase catalyzes phosphorylation, a phosphatase catalyzes dephosphorylation. And so see, this is an unphosphorylated form, right? So it's this form again, and in this case, then remember originally, that's not the binding site for potassium in this conformation. So now it ejects the potassium and get back to the original form. So all of this has to do with changes in conformation, whether it's changes in conformation of the binding site specifically or of the entire molecule. And this goes back to the importance of folding, right? That's why folding is so important. We good? Not so bad, right? Yes, Sam?
Changes of conformation of the binding site and the molecule.
Right. So conformational change can be changed in anything. Well, as long as it relates to the protein. It's a change in any domain, in any parts of the protein.
They use potassium, I'm guessing.
Okay? So that's one means to control structure function. Right? One example. Let's look at more ways to control. Again, now we talk about structure function, right? So conformational change. How do you induce more conformational changes? Yes, ma'am. Or, no, okay. Oh, yes, sir.
So, that phosphate group is already in the cytoplasm.
Phosphorylation is the phosphate group came from the ATP, but right.
It takes the sodium ions to enter that pump to activate the binding of phosphorylation. The binding of that phosphate.
The binding of 3, not 2, not 1, but the binding of 3 sodium ions causes the entire pump to change conformation, and the change in conformation then is recognized by the kinase. And now the kinase then catalyzes phosphorylation. Right. So the kinase itself. So even for enzymes, like I said, in order for an enzyme to carry out a reaction, to catalyze a reaction, binding still must occur, right? A kinase has to have the proper binding to the substrate, in this case the pump. Okay? So binding is it's again all about size, shape, and charge. I can't stress that enough. It's all right. Okay? So cell can be controlled by protein-protein interaction, or by can a protein function can be controlled by another molecule, right?
Okay.
But not by covalent modification, but by binding. Does that you
Yes.
see the difference? So like phosphorylation causes a change in the structure, conformational change, but that's covalent modification. You can actually have control of protein activity by having regulatory proteins or regulatory molecules binding to the protein, right? To either activate or inactivate the protein.
That's right.
and still activating and inactivating the protein still has to do with conformational change, still structure and function. So in this case, a protein of this conformation right, is inactive. In order for it to be, and it's inactive is because the catalytic site here is not in the right conformation, right? So this, let's say, is a not that, but this catalytic site is not in the right conformation, and let's say this is an enzyme that has to do with glycolysis. Okay, so glycolysis means that this enzyme has to bind to glucose. So in order to get that to be in this conformation, right, so that it can bind to glucose, then a regulatory protein must bind to that site to induce conformational change of the whole thing so that this enzyme would shut and therefore have the proper binding site for glucose. So therefore, regulation of whether this enzyme is active or not depends on this regulatory protein right here that looks like an arrow, the green arrow. So in the absence of this regulatory protein, no, you don't have activity. In the presence of this, you have a lot of activity. So and the next thing goes back to remind you about chemistry, right? How come this is working? Because what is that big concept about in chemistry, in organic chemistry that you learn? Why is it that this doesn't have a regulatory protein, an activator, and yet it's still working? Why is that? Anybody can tell me? I'll give you 1,000 virtual points. Getting there.
Oh, it's the.
What is a function of an enzyme? A function of an enzyme is to decrease the activation energy, but even if you don't have an enzyme, can a reaction still occur? Yes. It's going to take forever to occur. That's why this, right? That's why even if you don't have a functional enzyme, even if you don't have a functional regulatory protein, this may still occur. And it kind of has to do with equilibrium, where the more of something you have, the more likely that it happens, right? So therefore, even if you don't have the regulatory protein, yes, you could still have the reaction. It's just going to take a lot longer, right? Whereas if you have that regulatory protein, that would always cause the enzyme to be in the right conformation to then carry out glycosylation. So I'm forcing you to remember stuff from before. The reverse is also true. So that green thing is an activator. So that's how a protein is activated. How about a protein that is always active? You can turn it off by an inhibitor. Same idea. Same idea is that the activator and inhibitor, all they do is they bind to the protein to cause what? Conformational change. Right? And a conformational change, if it changes into a conformation that is active, then that's activating. If the conformational change is into a form that is inactive, then that's inhibitory. That's all that is. So the two slides are the same thing. Talks about the same thing, conformational change. And the regulation of conformational change has to do with the interaction with another molecule. It has nothing to do with modification of that particular enzyme. Okay? Yes, ma'am.
And that other molecule, is it usually a protein, or can it be any
It can be
type
just
of-
about anything. So a perfect example with glucose in a roundabout way, cyclic AMP actually controls. So the amount of glucose and ATP are usually inversely proportional. If you have too much ATP, you don't need a lot of glycolysis. You don't need glycolysis. If you don't have a lot of ATP, then you need a lot of glycolysis, which means you need a lot of glucose. But then the amount of ATP is also inversely proportional to cyclic AMP, because cyclic AMP comes from ATP. So then the cyclic AMP then can be, it's not a protein, but it can regulate. So that's why I say regulatory molecule instead of protein. All right, so I'm going to beat this to to death because we already talked about phosphorylation, already talked about covalent modification, and when covalent modification occurs, that changes the conformation, and that either turns on or turns off in a protein. Okay? So, and a perfect example, and we'll talk about phosphorylation and dephosphorylation many, many more times. The idea is that phosphorylation is a process by which a phosphate group is added. Dephosphorylation is a process by which a phosphate group is removed. Phosphorylation is catalyzed by kinase. Dephosphorylation is catalyzed by phosphatase. I already said that earlier. Okay? So here's where a lot of people make a mistake, and I want to right off the bat tell you that phosphorylation and dephosphorylation, we say that they regulate protein function. So it's not always true that phosphorylation activates a protein and dephosphorylation inactivates. Right? Sometimes you have phosphorylation that inactivates a protein and dephosphorylation that activates. As in here, so phosphatase can either turn on or turn off or turn on, and kinase can either turn on or turn off. So we say, therefore, phosphorylation and dephosphorylation regulates protein activity, depending on what? Depending on the type of enzyme, the type of protein that's there. Okay. Perfect example would be lamin, for instance. Lamin is a subunit for something called the nuclear scaffold, which is an intermediate filament. And lamin by itself doesn't work. It has to undergo polymerization, and this is an idea of polymerization leads to functions of a polymer. Now, lamin in the unphosphorylated form is allowed to bind and form the nuclear scaffold, which then work as a polymer. In order to inactivate that nuclear scaffold, guess what? Lamin has to be phosphorylated. So that's an example of phosphorylation actually turns off the function of a protein and not turning it on. Okay? So again, one more time, phosphorylation and dephosphorylation regulate protein function. Either one can turn on, either one can turn off protein function. But how does each do it? How does each regulate protein function? By causing conformational change. In addition to that, any and all covalent modification would induce conformational change. Again, any and all covalent modification will induce conformational change. We talked about glycosylation. Is there any more that we need to talk about? Yeah. We talked about glycosylation, right? Glycosylation occurs either in the ER or lumen of the rough ER or the Golgi apparatus. And you can see here each color is each thing here, cyclic structure here is a simple sugar. It's a simple monosaccharide, and so they can be built in that they can form a complex carbohydrate that way. Okay, and you notice that this is in the ER lumen, and this is in the cytosol. And eventually, if this were a membrane protein, it will come out that this is the extracellular matrix, and this is still the cytosol. Okay, so the carbohydrate residue never sticks into the cytosol. So that begs the question, and I know you're thinking it, but nobody wants to ask is, why? Why do you think? Why do you not want, if you were to create this, why do you not want carbohydrates in the cytosol? What is the property of carbohydrates? I'm sorry?
The sugars that you have.
When you put sugar in water, what happens? It's soluble, so that means that it's polar, right? So it's polar.
The polar,
What happens? You've probably done this experiments either in high school in Bio 1 or Bio 2, or you might have done it at home when you cook. How do you dry? Let's say salmon. How do you dry salmon? You put salt or sugar. You can't put sugar. Sugary salmon doesn't taste very good, but you can put sugar, right? Salt, sugar, anything that's soluble. How does that dry the salmon? What does the salt or sugar do?
Oh, draws the water.
Sucks out, draw out all the water. That's what happens with carbohydrates in your cytosol. It'll just allow all the water molecules to surround it, and then that just causes the cell, the inside of the cytosol, to be dehydrated. You don't want that, right? So therefore you okay. So that's why you don't want carbohydrates to be in there. Sodium or any type of salt needs to be of a certain level also. That's why you have to regulate the amount of salt in right? But then you can say the same thing with the carbohydrates being on the surface. So that, actually I can tell you that actually is, you want the carbohydrates to be on the surface. Why is that? If you have a cell sitting there, what do you want? What do you say?
Signaling?
Outside of that, we're still talking about water. What do you want? What do you want the cell? You want the cell to be well hydrated. Right? You want the cell to be well hydrated. You want the cell membrane, your cell membrane, to be covered with a thin layer of water. So even if it's sitting there in a condition where it's not bathed in water, at least it has a thin layer of water. Okay? So then that's why all your cells has a thin layer of carbohydrates on the outside. Okay? And so one of the functions, in addition to signaling, but a huge function is to keep your cells hydrated. So that's the reason why, again, why you will never see carbohydrates sticking into or in the cytosol. Okay? Another type of covalent modification is the addition of lipid residues. There isn't a name for it, it's just addition of lipid residue or lipid linkage. And so what is the property of a lipid fatty acid mainly? Is it hydrophobic or non-polar? Right? Perfect to exist in a membrane. So let's say you have a soluble protein. Right? This is a soluble protein. It doesn't have a membrane domain. But in order to get that soluble protein to interact or to be embedded into the membrane, then you add a lipid residue, and then that lipid residue can insert in. So an example of this would be a soluble protein that is synthesized in the cytosol and never make it into the ER, right? So because it doesn't have a topogenic sequence. So there isn't a sequence that tells the cell to insert it, the machinery to insert it into a membrane. But this soluble protein needs to be membrane bound. So in the cytosol, there's an enzyme there are many enzymes that allow for lipid residues to be added to the protein, and therefore this protein now is membrane bound through the lipid residue, mainly fatty acids. And fatty acids structurally looks similar to an alpha helix anyway. Okay? So addition of lipid residue. And we'll talk more about lipid residues. You've heard things about lipid. And by the way, proteins with lipid residues attached are called lipoprotein. Okay? So you've heard a lot of things about lipoprotein. For instance, what have you heard about lipoprotein? What are some of the examples of lipoprotein that you've heard of? LDL. LDL is low density. The last L is lipoprotein. HDL, high density lipoprotein. VLDL, very low density lipoprotein. So the L in the last L in LDL, HDL, and VLDL stands for lipoprotein. So these are proteins that were made soluble and then have lipids attached to them. Not all lipoproteins become membrane-bound proteins, right? Not all of them. Like LDL, HDL, VLDL, they're not membrane-bound proteins. They're signaling proteins, a lot of them. cholesterol. Okay. All right, so you want more complexity? Let's have more complexity in terms of covalent modification. So let me draw back. So let's see, B. So all of this, the B part here has to do with covalent modification, right? So phosphorylation is covalent modification. Glycosylation is covalent modification. Addition of lipid is covalent modification. Acetylation is covalent modification. Ubiquitination is covalent modification, right? They're all covalent modifications because these, all of these things involve the addition of a small group of molecule connecting to the protein by a covalent bond. So far, so good. So far, we're just talking about them in very simple terms. Okay, and not all proteins are like this. And in fact, this is on the other end of the spectrum in terms of complexity. There's a protein called P53 that can be modified multiple times with multiple different types of covalent modification. In fact, phosphorylation itself, it can be modified 10, 12, 13 different times at different places, right? And addition of acetylation can occur, ubiquitination can occur, right? And when we talk about covalent modification has to be done on at very specific areas within the protein to form what? To form a pattern of covalent modification. That word again, pattern, right? So P53 is a very important protein that we'll talk about. It's a very important protein in general when it comes to studying diseases. And depending on the type of modification, P53 will never ever have all of these modification occurring at the same time. It will always have a pattern of modification. And depending on the pattern of modification, the code, right? Pattern of modification or code, then that leads to different activities, right? By the same protein. So this one protein, the reason why P53 can have so many functions is because of the different patterns of modification, the different codes that it has. Okay. So again, this is on the more complex complex end. But even when you talk about something like glycosylation, when you take biochemistry, or if you have, that's great. But I don't know if you learned that in micro, but glycosylation, you know that enzyme, what is it called? The enzyme that catalyzes the conversion between glucose and fructose, but can also go the other way. So one enzyme has actually two functions, right? Two opposing functions. And the reason why that one enzyme has two opposing functions but never ever carry out opposing functions at the same time is because the protein, that protein has to have one conformation or the other conformation. And how it has one conformation and change to the other conformation is by having multiple covalent modifications. Right? So by having a certain pattern of covalent modification. What is that? Anybody who's taken biochemistry, what is that called? The fructose phosphatase and something like that, or glucose kinase or something like that. Whatever you said. Yeah. Right? If you remember your biochemistry, that's it. And I actually met the guy who discovered that protein. Not a very nice human being, but very smart guy. Very smart guy. But then that's what I say about me. Not that I'm smart, but not very much. I will repeat this. This is yet another example of proteolytic modification, right? So that's another type of modification. Don't worry too much about memorizing this, but when we talk about apoptosis, that many of these enzymes that would carry out the that would lead the cell to die by apoptosis, right, are found in both the they're all found in both in inactive form and in an active form. And in order to activate this, each of these enzymes have to be cleaved. These enzymes have to undergo proteolytic modification. The thing about proteolytic modification is that there's no undoing it. So these enzymes cannot get inactivated again. So they're formed, they are synthesized in the inactive form, become activated through proteolytic modification, and then they get digested they get ubiquitinated and then delivered to the proteasome and then get degraded. Integration of information again. So this is to answer the question of what other protein besides misfolded proteins can be degraded by the proteasome. A caspase, right? So an active caspase is active until it's no longer needed, and then it's degraded. Until, of course, the cell would die, and then there's no need to degrade anything because it's just, right, the cell is dead already. Okay? We'll come back to talk about apoptosis, the whole pathway, later on in about the fourth set of exams. Okay. This is slightly even more complex. So we're talking about regulating a protein so far of directly regulating the protein function. But now we're talking about regulating a protein function by regulating another protein, which in turn regulates. So it's a series of regulation that leads to the final regulation of your protein. Okay? And again, we'll just look at this in general, and then we'll go into a lot more specific details later on. Not in this set of exam, but later on. Okay? For instance, this is a receptor. We talked about ligand and receptor, signaling molecule and receptor. So a signaling molecule binds to the receptor. Okay? The binding of, before the binding, you notice these two molecules aren't even touching each other. The binding of the ligand or signaling molecule causes these two molecules of receptor to undergo conformational change, and then after the conformational change, they actually can interact with each other. So far, so good. Okay, and it is only through a process called dimerization, because they form two dimers, right? Polymerization is a formation of a polymer. Dimerization is a formation of a dimer. Trimerization is a formation of a trimer, and so on and so forth. Okay? So you notice that, and then in the dimer form, now this dimer can now undergo phosphorylation of itself. So can undergo one molecule phosphorylates the other, right? It's cross-phosphorylation. There's another term called autophosphorylation. So here's the difference. Cross-phosphorylation means that if you have two things that one phosphorylate the other and cross like this, that's one way to look at it. But if you were to look at the dimer as one entity, then in fact the dimer phosphorylates itself. So in fact this is saying cross-phosphorylation. We took this from a book and it's not quite correct. This is actually, if you cross out cross and you write autophosphorylation, this is a better example of autophosphorylation. Later on, much later on, we'll look at a true case of cross-phosphorylation. This is more of autophosphorylation, okay? But you can see why the book calls it cross-phosphorylation, right? Because okay. So here's how. So there you go. This is a series of modification that leads to autophosphorylation, right? And then the autophosphorylation, the phosphorylated dimer now now is active to do something else. Does that make sense? So these two molecules all of a sudden cannot be phosphorylated, right? It requires the binding of a protein to cause dimerization so that autophosphorylation can occur, so that the autophosphorylated dimer now can have can be active. Yes, sir.
So the binding of the single molecule indirectly causes phosphorylation.
Right. Through this series of events. So binding here causes conformational change, which leads to dimerization. Dimerization then leads to more conformational change, which allow the dimer to undergo autophosphorylation. Autophosphorylation leads to even more conformational change, now allows the phosphorylated dimer to be active to do something else. It just happens that this phosphorylated dimer is also a kinase to phosphorylate other proteins. And we'll look at this pathway a lot closer later on. Okay? So we call this, there has to be a critical binding partner to get everything jump-started. Sequestration. This is, I just have to admit to you that this is probably one of the most difficult concepts, at least when it comes to taking the exam, for students to understand. Okay, so I won't stay on here for too long, but at least I'll try to explain it. That sequestration. What is sequestration? We hear this in politics, right? What is to sequester? To hold back, to store. Right? That's what sequestration is to store, right? So sequestration of effector molecules meaning regulatory molecules. A lot of effector molecules are metal ions. So in a way, if you were talking about metal ions, we would say that sequestration of metal ions can be useful in the activation of proteins. Okay? How does that happen? How does that work? For instance, if I remind you about like a cellular respiration, right? During cellular respiration, electron transport chain, what happens, not at the end, but during the electron transport chain, what occurs? Protons get stored into the intermembrane space in the mitochondrion, right? That's sequestration. During electron transport, protons are sequestered in there. To do what? To build a concentration gradient. You see that? Sequestration leads to concentration gradient. When there is a sufficient concentration gradient, so the gradient gets steeper and steeper and steeper, at some point, right, then protons would move down the concentration gradient, for instance, moving through a proton pump or the ATP synthetase, right? That then activate the ATP synthetase to carry out the conversion of ADP into ATP. So that's how sequestration of ions leads to the activation of an enzyme. And there are many more examples like that. So the example that we looked at in terms of sodium-potassium ion pump, the same thing, right? Sodium ions will not bind to the pump unless there's a sufficient concentration gradient. And so therefore, sodium needs to be pumped in first to create that, so to be sequestered, right? To create that concentration gradient so that it goes the other way. And then there has to be a sufficient amount of potassium on the other side. Well, actually, no, sodium, potassium pump. No, it doesn't work that way. I'm sorry. That goes against the gradient, so never mind. Cross that out. Okay. So, proton pump, ATP synthetase is a good example. Forget about the sodium, potassium. Some of you are going to come back to me and say, hey, okay. Remember ABC transporter? Remember 77 members of the family on the first exam, where there's that what species is that has 4,000 genes, and there's a member that has 77 members of the ABC protein pump, right? That's it right there. Okay. Go back and I'm not requiring you to memorize that. I'm just linking that it comes back again. Okay. Sequestration of calcium is also important. Calcium is either sequestered in the ER lumen,
It's like guitar.
in mitochondrial matrix, or in the extracellular matrix. Okay. So in the cytosol, usually you don't want calcium ion. So if you have calcium ion in the cytosol, guess what? It gets pumped into these three places. So even so, we consider the extracellular matrix as a storage space. And when you want calcium ions to be in the cytosol, when you want a protein that requires calcium ion to be activated, right? So by sequestration of calcium ions before activation of protein, once the sequestration creates a high enough concentration gradient, and then and only then calcium ions are allowed to move into the cytosol. But that's regulated, right? Having a concentration gradient alone doesn't mean that calcium will flow into the cytosol. That process has to be activated as well, regulated as well. But again, even if you regulate the flow of calcium ion, but if there isn't a concentration gradient, then calcium ions will still not flow into the cytosol. Does that make sense? And that actually point is also important when we talk about contraction. Okay, that the concentration gradient of calcium is really important to keep contraction going, and this is why your muscles get fatigued because of this lack of concentration gradient of the calcium ions, which means that there's a lack or let's say lack for now of sequestration of calcium ions. You see how sequestration of ions are important? Okay? So we'll come back to this idea again later on when we talk about contraction. Okay, more. Almost there. I forget how many more slides there are. So, cooperative coupled or coupled binding, meaning that binding to one site. So hemoglobin, you must have heard the story somewhere else. Hemoglobin has 4 binding sites for oxygen. Let's just look at oxygen first, right? Let's say this is 1, 2, 3, 4. Site 1, site 2, site 3, site 4. Right? Oxygen will not bind to site 4 or will not bind to site 2 unless site 1 is bound by oxygen. So oxygen binds to site 1 first. That causes conformational change that now site 2 is open for binding. Site 2 binds to oxygen, that causes conformational change so that site 3 can bind to oxygen, and so on and so forth. Right? So the binding of one site regulates the binding of another site. That's called cooperative or coupled binding. So oxygen will not go into site 3 and start binding. The reverse is true. In fact, the same binding site can bind for carbon dioxide. So carbon and oxygen fight it out to bind to the first site. If carbon dioxide binds to the first site, guess what? Then the second site has a conformational change that would only bind to carbon dioxide and not oxygen. Okay? And the third molecule, which is the dead molecule, carbon monoxide, same thing. In fact, carbon monoxide has I believe, a higher affinity for that first site. So if you have a slightly raised carbon monoxide level, it doesn't have to outweigh carbon dioxide or oxygen, you're in trouble. Okay? So that's cooperative. So the binding of one side then regulates the binding of another side. Kind of sort of similar to what we talked about earlier, allosteric binding, right? Where the binding of one site by a regulatory protein then can allow for the active site to be actively inactive, to regulate the active site. Okay? Regulate protein function by blocking the binding sites. So in this case, in this specific example, we're looking at a protein being in the form of a polymer. So a polymerization would then induce function. But the polymer itself, its function can be inhibited by binding, by blocking the binding sites. So again, in terms of contraction, what happened is that tropomyosin is that binding site blocking protein. So tropomyosin blocks the binding site here. Okay? And only if tropomyosin were uncovered will that binding site in here be exposed, right, to the myosin head, and then you have contraction. So as long as tropomyosin blocks that binding site, then contraction can occur. So then blocking of the binding site by a protein is a way to regulate protein function. Okay. And by the way, these various means of regulation, they can work along with one another. You already saw that earlier, where binding by a signaling molecule then induces dimerization, which leads to phosphorylation, which then activate the kinase, right? So in that case, you're talking about allosteric interaction, allosteric control, plus somewhat polymerization control in terms of dimerization, plus covalent modification, right? So you have all of these types of controls working together sometimes. So they don't work independently. Sometimes they work together. Okay? Polymerization, just to repeat another idea, is during cell division, and you remember the cell pinches, right? The cell pinches through the formation, so through the polymerization, and then through the assembly and then disassembly of the microfilament. So assembly and disassembly of microfilaments, which is a polymer, right, controls cell division, controls at least the cytokinesis. Okay. So it's just an example of, again, how formation of a polymer as well as the breakdown of a polymer can control activities. Scaffolding. What is scaffolding? Scaffolding is when a molecule or protein is put on top of one another, right? That's scaffolding. So let's say, in this case, here's a green protein. It needs to recruit the brown protein, but the green protein can't recruit the brown protein directly. So what happened is scaffolding has to occur, so that the green protein recruits the blue protein, which recruits the purple protein, which recruit the brown enzyme. At the same time, another protein at the same time, the unwanted protein has to be recruited to that original green protein, so that ubiquitination can occur. And this leads back to how unwanted proteins are destroyed. And ubiquitination then, this ubiquitinated protein now gets released and get delivered to the proteasome. So scaffolding again is the piling up of protein, okay, of related protein. Same idea is during the synaptic signaling. During synaptic signaling, right, a neuron interacting with so the signaling cell interacts with a target cell, right? Presynaptic cell versus postsynaptic cell. But we talked about the fact that the neuron has to make connection with the postsynaptic cell or the signaling cell, and then only very specific cell. So that's the idea of the DS-CAM protein, remember that? So the adhesion, right? So then there's specificity in terms of signaling cell and target cell, not only in terms of cell, but in terms of the specific signal And when communication occurs, you don't really have just one signaling molecule being bound by a receptor. You have a whole complex of proteins that are involved. How do you keep these complex of proteins in place? So let's say this right here is it is not to say, but postsynaptic membrane. So this is a target cell. Here's a signaling cell up here, right? So the signaling cell is going to produce a neurotransmitter, which then is recognized by, let's say, these receptors. But in addition to these receptors, all of these proteins have to be related. Okay? So all of the related proteins that carry out the same functions will be gathered in the same place. How are they gathered in the same place? By this scaffolding protein. So this scaffolding protein would have multiple binding sites to bind to all of the related proteins to keep them in one place. What does that have to do with anything? Okay, so a little bit about physiology. You know that to activate a cell to carry out action potential, potassium voltage gated potassium ion channel had to be open, right? And how is it activated to be open? By activating the receptor. So by activating this receptor, right, then the activated receptor or receptors, so sometimes multiple receptors have to be activated. So, well, okay, I'm not going to pile on to different ideas. Let's just keep it in terms of the receptor has to be activated. Activated receptor then in turn activate the potassium ion channel. So the potassium ion channel cannot be activated because it itself is not a receptor, so it can't be activated by a ligand. Right? So a ligand will activate a receptor, which in turn activate the potassium ion channel. So therefore, the potassium ion voltage-gated potassium ion channel always needs to be in the same area, needs to be connected with the receptor. And how is it connected with the receptor? By scaffolding. Right? So scaffolding, again, the idea is to gather up all of the components in one place. Basically, in layman's term, I guess, or in simplified term, definition of scaffolding is to gather up all of the related protein in one area. You gather up these proteins either by having one scaffolding protein with everything binding onto it, or you pile up proteins, right? One protein leads to the binding of the next, which leads to the binding of the next, which has to do with cooperative binding or allosteric binding. Right? You see that? You see that it does have something to do with that, right? You sure this is the last slide? Oh no. So more about scaffolding is scaffolding can also help, again, the same idea is help gather all these enzymes, but all of these are enzymes that have that one may act. So in a pathway, sometimes you have an enzyme, right? You have a pathway, a series of reaction, right? So if you activate one reaction of a pathway, if the other reaction isn't in the same place, it'll take some time to find that other substrate, right? To lead to the next reaction. But if you line up all the enzyme and substrate in a row, then as soon as you activate the very first one reaction, then all the other reactions will occur quicker. So we call this hardwiring. So scaffolding is a way to hardwire, like hardwiring electrical stuff in a building. Hardwire so that when you flip the switch, all of this turns on, right? All of the lights turns on. So once you flip that switch, the whole pathway occurs very quickly. So it allows for speed. And sometimes scaffolding also allows for not a whole reaction, but for multiple activity to occur that have to do, so not necessarily a pathway, right? But sometimes you in order to undergo glycerol synthesis, then multiple activities have to occur at the same time, right? In order to activate, so you would like to turn on one thing and then have all these activities occur at the same time. You don't want to have to turn each one of them on, right? You don't want a switch for each of these light bulbs. You want one switch that would turn on all the light bulbs, okay? So that's in essence what that means. Exactly. Well, I have 30 seconds.
Is that it?
Good luck, everyone.